Hello, and welcome to this part of the FAST tutorial series. In today's video, we'll be looking at how to use FAST's inbuilt plotting module to visualize the data that we've extracted from the tracks. So, as usual, we begin on the home panel screen, and you should be able to see that this plotting button has turned green after you've completed the tracking module. So to start the plotting module, as usual, we just click this button, and we have this screen pop up. And this contains this on the right, which is going to be where our plots go, and this on the left, which contains the settings. Now, to demonstrate this module, I'm going to be using the same dataset as we were using in the previous demonstration, the overlays module. This is a dataset consisting of these bright puncta that appear within cells and move around a bit and then disappear. And we have two populations of cells here. We've got Pseudomonas aeruginosa and Vibrio cholerae. And these are both labelled with different colours of fluorescent proteins. So in this case, this is channel 2. This is in Pseudomonas aeruginosa. And then there is a second channel, channel 3, which contains the red protein. That's Vibrio cholerae. So the plotting module contains basically two different types of plot. The majority of the plots collate data across the entire set of tracks, and then we have these two types of plot here, the cartouche and the Kymograph, which show data for only an individual cell. Now I'm going to start off by demonstrating the Kymograph. I won't be demonstrating the cartouche option here, but you can find more details on the wiki. So this pops up, this plot, and this is essentially a classical Kymograph. So we have along this axis here, we have the distance from the cell midpoint, so this is one pole and this is another pole, and then we have time over here. What this is doing is it's taking each cell and then averaging across its narrow axis. So what we can do is, instead of just looking directly at and trying to find cells of interest, we can do something that I mentioned in the previous video, which is to open the overlays module, select the channel of interest, click this show ID button, and that allows us to find where specific cells and specific tracks can be found. So let's just move through to the middle. Let's say that we're interested in looking at this cell here. We can then click show ID and see that it has the ID 188. Then we can close down the overlays module, open the plotting module, select Kymograph again, and then type in 188 into the track ID box, and then you can see that this is the data corresponding to that cell of interest. There's another option here which allows us to equate lengths of different objects. So if we've got cells that are growing over time, then it will expand out those shorter cells at earlier time points so that they every sample has the same length, but obviously clicking that in this case doesn't do a great deal. So you can also choose different color maps, again similar to the previous video. These are the built-in MATLAB options, so we can have, for example, copper, and then that looks like much more sepia tinted. Now for the second class of plot, which is the one that collates data across all different tracks, I'm going to be using this joint distribution plot type. Again, I'll just be demonstrating one example, but you can find the details of the other plots on the wiki. And what I have done before this video is I have split all of my tracks into two separate populations. So this is similar to what we were discussing in the previous video, where we have splitting of different events. Now we're labeling tracks according to the population that they belong to. And in this case, I've split the tracks according to the species. So half of the tracks approximately correspond to Pseudomonas aeruginosa, and half the tracks correspond to Vibrio cholerae. So just here, I'm going to choose the two different variables that I want to plot against each other. So that's going to be the average of channel 2 and the average of channel 3. And then I can choose which subpopulations I want to display on these axes. So here I'm going to choose population 1, population 2. And you can see that we have this nice clustering of these two separate populations on these axes. And this is actually how these two populations were split. So basically, when this channel 2 mean is high, that means that the green protein is highly expressed, and that suggests that it is a Pseudomonas cell, whereas when channel 3 is high, that indicates that there's a large amount of red fluorescent protein present, and that corresponds to Vibrio being present. And this here, split for population tracks, does exactly this, so you can see that this is the same plot that we have generated here. And you can find this again on the wiki in the post-processing toolbox. So once we've done this, we can choose to use different colors to represent our different populations. So for example, purple and green. We can also add a legend to these axes. We can choose the names of our different populations. So population 1 is our Pseudomonas, I believe. And we can also choose the name of population 2, which is our Vibrio. And you can see that we've updated this legend with those two different names. We can also, in this particular case, in this additional options box, we can choose to show the correlation coefficient between these two different variables, which isn't particularly informative for this particular pair of variables, but obviously depending on what you're interested in looking at, this could be very useful. We can also plot fits, so regression analyses of these two different populations. And we can also mix in the entire population as well, if we were so interested in doing. I think this is a case of Simpson's paradox, but that's a conversation for a different time possibly. So once we're happy with the axes and how they look, we can finally export the plot. To do that, we've got three different options. We can either say save axes as a .tiff, and if we do that, then we will have in our root directory, we'll have this .tiff exported plot.tiff file produced, and you can see that that contains the axes as they appear in the module as a bitmap. We can also save as a .fig, and that will create a MATLAB figure which we can then open in MATLAB, and that's called exported plot.fig. And so we can then edit this using MATLAB's inbuilt figure editor. So say, for example, we wanted to make these points all much bigger. Then we would go to figure properties and double click these points, and then we could increase, say, this to 15, and that would increase the size of all of our points. If even that isn't sufficiently flexible for our needs, we can also choose to export our figure data directly. That will create a new .mat file, which is this plot data export.mat file. And we can open this now directly in MATLAB. And then we can look at our different populations as cells within this plot export variable. So for example, if we choose to look at the second field of plot export, that corresponds to population 1. The first cell corresponds to the entire population, so that isn't included here. So when we look at this second cell, we can see the properties of this population 1. This contains data for our x and y variables in the plot figure. So here we have channel 2, so that's our x variable, and here we have channel 3, that's our y variable. And then we also have these numbers here which correspond to these numbers up here, which are the correlation coefficient and the significance of the correlation between these two different variables. And with this exported data, we can then go on and produce more fancy looking plots using this data directly. So it really does give you a great deal of flexibility in how you can choose to arrange your data. So that is the end of the FAST tutorial. Thank you for joining me in this discussion. I hope that you find it a useful tool for your research.